Good morning everyone. In the previous video we learnt oxidation of fatty acids, that too oxidation of saturated fatty acids, which consists of even number of carbons and also odd number of carbons. How they get oxidized and how the energy production takes place, that part we learnt. In this video we will see oxidation of fatty acids of unsaturated fatty acids, and also we are going to see alpha oxidation and omega oxidation. Oxidation of unsaturated fatty acids. Before going to this, I just mentioned what is saturated fatty acid and what is unsaturated fatty acid. Saturated fatty acids consist of single bond, whereas unsaturated fatty acids consist of double bonds. By the oxidation of unsaturated fatty acids, we will get less energy when compared to the saturated fatty acids. The reason for this is saturated fatty acids, since they possess double bonds, the breaking of the double bonds is difficult compared to the single bond breakage. So that's why they provide less energy. Unsaturated fatty acids provides less energy compared to the saturated fatty acids. So that means presence of double bond poses a problem for beta oxidation to proceed, and this problem is overcome by two additional enzymes. Those are nothing but isomerase and epimerase. With the help of these two enzymes, isomerase and epimerase, it becomes somewhat easy and gives some energy with unsaturated fatty acids. And this is regarding beta oxidation in peroxisomes. So beta oxidation in mitochondria we have learnt, but here the beta oxidation in peroxisomes. What are peroxisomes? Peroxisomes are present in most of the eukaryotic cells, and they are the cell organelles, and it occurs, beta oxidation in peroxisomes occurs in a modified form. Here we won't find any ATP synthesis, because reducing equivalents whatever are produced, they do not pass through the electron transport chain. The reducing equivalents, when they pass through the electron transport chain, they generate ATP. But here in peroxisomal beta oxidation, they don't pass through the electron transport chain, so that there is no ATP synthesis. And what is going to happen instead of entering the electron transport chain? These reducing equivalents are directly handed over to the oxygen. That results in the formation of hydrogen peroxide. And this hydrogen peroxide is toxic to the cells. And how about this hydrogen peroxide? If it remains, it is going to cause toxicity to the body. So that a catalase enzyme detoxifies this hydrogen peroxide to water and oxygen. That is peroxisomal beta oxidation. This peroxisomal oxidation, it is induced by high fat diet and also administration of hypolipidemic drugs like clofibrate, etc. and this is regarding peroxisomal beta oxidation. And here I would like to mention one syndrome, that is Zellweger syndrome. It is a rare genetic disorder which is characterized by the absence of peroxisome in almost all tissues. So that is, when peroxisomes are absent, what is going to happen is long chain fatty acids are not going to be oxidized, which consists of more than 20. So these fatty acids are not going to be oxidized, so that they are going to accumulate in tissues, particularly in brain, liver and kidney. And when they accumulate in the tissues like brain, liver and kidney, they are going to disturb the cellular function and also causes serious problems soon after birth. And this disorder is also known as cerebrohepatorenal syndrome. So that is about peroxisomal oxidation. And now we will see alpha oxidation of fatty acids. So beta oxidation of fatty acids is the most common one, because mostly the fatty acids are oxidized on the beta carbon atom of the fatty acids. But alpha oxidation is also possible. Here what happens is the removal of one carbon unit at a time by the oxidation of carbon atom of fatty acid, and it is not going to involve the binding of fatty acid to coenzyme A, and no energy is produced in alpha oxidation. And another one is I would like to discuss here is Refsum's disease. Refsum's disease is also a rare severe neurological disorder which is characterized by the cerebral ataxia and peripheral neuropathy. And the patients of this disease, they accumulate large quantities of an unusual fatty acid, that is nothing but phytanic acid. This phytanic acid is derived from phytol. Phytol is a constituent of chlorophyll, which is found mostly in the plant foods, and also it is present in the milk lipids and animal fats. This phytanic acid cannot undergo beta oxidation due to the presence of a methyl group on carbon 3, and this fatty acid undergoes initial alpha oxidation to remove alpha carbon as carbon dioxide, and that is going to be followed by the beta oxidation. So this is about the Refsum's disease. And this Refsum's disease is characterized by a defect in alpha oxidation due to the deficiency of the enzyme, that is phytanic acid alpha oxidase. Hence this phytanic acid cannot be converted to a compound that can be degraded by the beta oxidation. First it has to be converted to a compound, and then that phytanic acid can be converted to a compound which can be degraded by the beta oxidation. But here, with a defect in the alpha oxidation, deficiency, because of the deficiency of this alpha oxidase enzyme, that phytanic acid is not going to be converted to a compound which can be degraded by the beta oxidation. And the patients who are suffering from Refsum's disease, they shouldn't consume diets containing chlorophyll. For example, green leafy vegetables. That is about the alpha oxidation. And coming to the omega oxidation, it involves hydroxylation, which is followed by the oxidation of omega carbon which is present as a methyl group at the other end of the fatty acid. It takes place usually in smooth endoplasmic reticulum of liver and kidney of vertebrates. And this reaction, omega oxidation reaction, requires cytochrome P450 NADPH and also oxygen. It serves as an important alternative to defective beta oxidation pathways. And some research studies have shown this omega oxidation can form glucose from succinyl CoA in times of starvation and also diabetes. It is important for pathway for the production of pheromones. So that is about omega oxidation. So in this video we have seen the oxidation of unsaturated fatty acids. Unsaturated fatty acids is also possible, but it gives less energy, and by having the double bonds it is somewhat difficult, but it is possible with the enzymes like isomerase and epimerase. And coming to the peroxisomal beta oxidation, peroxisomes are certain cell organelles, and the reducing equivalents produced here, they are not going to enter the electron transport chain, but they are going to directly combine with the oxygen and forms a hydrogen peroxide. Of course, this hydrogen peroxide is toxic to the cells, so that it is going to be cleaved by the catalase enzyme, antioxidant enzyme, and it forms water and oxygen. Then Zellweger syndrome, it's a rare genetic disorder which is characterized by the absence of peroxisomes in almost all tissues. When peroxisomes are absent in the tissues, what is going to happen is long chain fatty acids are not going to be oxidized, so that they are going to accumulate in tissues, especially in brain, liver and kidney, and that disorder is known as cerebrohepatorenal syndrome. And serious problems occurs in soon after the birth of a baby. And coming to the alpha oxidation, to summarize this alpha oxidation, removal of one carbon unit at a time by the oxidation of carbon atom of the fatty acid, and here it is not going to binding of fatty acid to coenzyme A, and here also no energy is produced. Because of the defect in this, Refsum's disease may occur. It is also a rare severe neurological disorder, and these patients accumulate large quantities of unusual fatty acid, that is phytanic acid, and this phytanic acid is usually oxidized by the enzyme phytanic acid or alpha oxidase. If that is not going to be converted to a compound which can be degraded by the beta oxidation, these patients will suffer from the Refsum's disease, and they shouldn't eat. The patients who are suffering from Refsum's disease shouldn't consume diets containing chlorophyll and green leafy vegetables. And the last one is omega oxidation. It involves hydroxylation, which is followed by the oxidation of omega carbon, which is present as a methyl group at the other end of the fatty acid. And it takes place in smooth endoplasmic reticulum of kidney and liver of vertebrates. And the reactions of omega oxidation requires cytochrome P450 NADPH and oxygen, and Some studies have shown this omega oxidation can form glucose from succinyl CoA in times of starvation and diabetes, and this is very important pathway for the production of pheromones. This is about omega oxidation. So along with the beta oxidation, omega oxidation of fatty acids is there, and also alpha oxidation of fatty acids is there, and along with the mitochondrial oxidation of fatty acids, some other organelles, cell organelles like peroxisomes also are capable of undertaking oxidation of fatty acids. So this is all about the oxidation of fatty acids. Thank you.